Yeah, no problem. Thanks, thanks, Dave. I'm Dave Barber. I work up in Yorkshire area for the Environment Agency. I work in the analysis and reporting team as a senior environmental monitoring officer, which is basically an ecologist covering all things ecology, from fish and inverts to algae and chemistry. Yeah, Martin, do you want to?
Well, yeah, I'll just say snap, I suppose, except that I work in the West Midlands area, but I do pretty much the same job as Dave, and we sort of cover very similar roles.
Okay, I'll kick off then. Well, good morning, everyone. Martin and myself are here today to provide you with an operational perspective of the Environment Agency's use of nutrient impact data, and that includes assessing the risk of eutrophication using the weight of evidence approach. So a couple of aims today. I'll let you read those rather than reading them out on the slide. But hopefully they set the scene and provide an outline of what we are and indeed are not covering today. So we'll be focusing on the operational detail, and in particular, as we're both ecologists, on the ecology data sets that we use. We won't be covering in any detail the strategic or legislative landscapes around nutrient management in the Environment Agency, other than to say the weight of evidence tool or approach that we're covering is used in both WFD river basin planning and also for identifying measures for water company asset investment plans. Okay. So just in summary, this talk today, I'm going to run through the eutrophication weight of evidence approach firstly, and then we've got a couple of case studies, firstly from the River Wharfe in Yorkshire, and then Martin's going to cover the River Wye, and then some conclusions and a Q&A session. So I'm sure everyone's familiar with eutrophication and the process of eutrophication, but it's effectively an ecological response in a water body. That could be a pond, a lake, or a river, to excessive nutrient loading, and you get prolific plant and algal growth, and that causes all kinds of ecological issues. The weight of evidence approach that we use at the Environment Agency is a decision support tool. It combines multiple strands of evidence around nutrient impact in a standardized manner, and it gives you a assessment of the likelihood of eutrophication or the eutrophication risk in a water body. And the outputs from the tool, it puts each water body into 5 classes. So it's done at the WFD water body level, I should say, and each water body gets classified against one of those 5 outcomes. Obviously, there's from very certain there's a eutrophication problem through to certain no eutrophication problem. And I should say there's weight of evidence tools have been developed for rivers, lakes, and transitional and coastal waters, but what we use and what we'll be talking about today in ANGA is the river tool. So we're going on. It's not moving on for some reason. So this is what it looks like. It's a great big spreadsheet, which might be quite visually wow as you first look at it, but it's quite user-friendly. What you do, each one of those lines is a water body, and you work along the spreadsheet. This is actually all one spreadsheet, but I've had to put it in two lots on the slide because it's so big. But you basically work through along the line and fill out each piece of information that's requested and answer the questions that are posed. And whirring away in the background are several matrices and weightings that effectively arrive at the final decision of certainty. What I'll attempt to do over the next few slides is try and break that huge spreadsheet down into just sort of outline the process of what's happening there. So this is a bit of an overview of the process. Initially, there's an assessment of the certainty of eutrophication based on the national WFD classification data, or the relevant classification data for a given water body. So for nutrients, That will be for macrophytes and diatoms for the biological tools, and phosphate for water chemistry. Obviously, we're working in fresh waters, so the assumption is that phosphate is the limiting nutrient in those water courses. That again gives this initial certainty of a eutrophication issue. We then are able to add in local EA area evidence, and that's broken into 3 sections, which I'll go through in a lot more detail. Pressure evidence, measured evidence, and other supporting evidence. And then that arrives at the final certainty of a eutrophication issue. It's not moving on. Okay, so if we look at this section first, the initial WFD assessment, and look a little bit more detail what's happening there. In this section, firstly, the macrophyte classification and diatom classification for a WFD water body are combined. The combined macrophyte and phytobenthos, another read diatoms there, that's another word for it, are then combined with the phosphate classification, element classification for the water body, and that then gives the initial view of the certainty of eutrophication in that water body. It's a little bit more complicated than just the combining, working along underneath this. So there's weighting applied based on the confidences of the classifications and where in the sort of distribution of probabilities and in that class, the confidence of class, the particular classifications sit for each of those. And also there's weightings applied between macrophytes and diatoms based on the river typology and which tools will do. So this is one of the examples of the matrices that sit below this. You can see this is an upland, low alkalinity river typology, and there's more weighting applied to the diatom tool in those river types. Okay. Moving on. So moving on to the area supporting the area supporting evidence that we can add in there. So taking that initial assessment here through stepwise these 3 different categories to get our final assessment. Each of those categories, the pressure evidence, other measured evidence, and other supporting evidence, has several strands that sit underneath them, and we'll go through all those. And each of these, you have to review the evidence that's available for each of those different strands and answer, is it showing evidence of nutrient enrichment or eutrophication? And it's a yes, no, or not assessed. And that cumulatively scores across and amends that initial assessment to get your final assessment of certainty. So your pressure evidence, this is around looking for 4 additional indicators of eutrophication risk. So, for example, we've got additional phosphate spot sampling data, and that'd be from non-WFD classification sites used for investigations. Also, if there's sewage works discharging into the water body or upstream, or if there's if your phosphate classification is either poor or bad, it all adds to that weight of certainty that you're going to have a eutrophication issue. The other measured evidence, again, is comprised of 4 strands, and this is local environment agency, again, non-WFD classification data. So this is from investigations. So this will be from additional macrophyte or diatom surveys that have been carried out in a water body for investigatory reasons. It could be from continuous water quality monitoring from our SOND program, showing diurnal variation in dissolved oxygen or pH. Again, measured excessive algal or plant growth. So that would be going out and doing an assessment of cover values and species. And phytoplankton blooms as well can all play into that other measured evidence category. And the final category, and the one to really focus on today and relevant for CaSTCo, I think, is the other supporting evidence. So there's 5 different strands to this. And what I'll do is jump through to the guidance for each of these different strands of evidence. And I'm sorry, this slide's quite wordy, but I think it's worthwhile having taken a bit of time to read all of those different strands of evidence and the guidance for those, because I think you'll see that there's scope for citizen science to play a role in all of those. So hopefully everyone's had a look at those, and we'll come back to that with a worked case study. So that is the weight of evidence tool. So just in summary, it brings together all the WFD classification data for a water body to give an initial certainty, and then that initial certainty is then amended by local area supporting evidence, and that can include external citizen science data in that process, and that then gives us our final certainty of a eutrophication issue. So I'm going to run through a case study on the River Wharfe in Yorkshire, which I'm sure everyone's familiar with now from previous webinars. But I will, just in case you're not, the River Wharfe, it's one of the rivers coming from the Yorkshire Dales. It's got its source just up to the northwest of Kettlewell, up in the Yorkshire Dales. It runs down through Wharfedale, going past towns such as Ilkley, Otley, Wetherby, Boston Spa, and has its confluence with the Ouse just to the southeast of Tadcaster. It's a river of two halves in many ways. It starts as your stereotypical sort of limestone upland river. Then as we come down, it gets wider and flatter, and we get more urbanisation and larger issues coming in particularly from sewage works and larger changes, and it ends up as quite a lowland wide river with sandy banks and meandering. You can see there at the bottom. And what I would say is, on many measures, it's a really good quality river. So this is the invertebrate classifications. It's actually high status all the way down from source to confluence. And this is supported by the ammonia and dissolved oxygen classifications. And the invertebrate tool is very much built around picking up organic pressure. And so we can see from organic pollution, we're not picking up a huge signature or an issue on the wharf. But what we know we do have problems with are nutrients. So this is filamentous algal growth over some of the substrate up in the mid to upper catchment. And whilst I'm not covering it today, obviously, it would be remiss not to mention on the wharf, we do obviously have issues with fecal bacteria loading. That's for another webinar, I think. But what I would say is it's worth bearing in mind that there's quite a lot of overlap when looking at nutrients and fecal indicator organisms, both in terms of sources, but also pathways to the river, whether we're talking diffuse agricultural pollution or sewage pollution in the watercourse. So the background to the wharf work was that we were tasked as the Yorkshire Analysis Reporting Team to undertake an assessment of the nutrient status of the river to support Environment Agency decision making going into PR24 and determining measures that were to go into the water company investment plans, and particularly as to whether the wharf was to get designated as a candidate sensitive area under Urban Wastewater Treatment Directive regulations. So the first part of obviously undertaking the weight of evidence assessment is to do the WFD classifications. So the first thing we did was to update all of the WFD classifications on those main river water bodies. And these are the results. They're ordered from upstream to downstream, working down that table. And then if you look across, we've got those nutrient sensitive elements, macrophytes, diatoms from 2 different years, and phosphate. And I think what you can see is macrophytes show an issue pretty much from source all the way down. This is largely driven by filamentous algal cover, such as you can see in the pictures associated with this slide. Diatoms show a similar deteriorating trend moving downstream, but to a lesser extent, and again a similar pattern in phosphate. But what you'll notice is there is mismatches across each of those water bodies, across the different evidence types, and This is common. We see this a lot with nutrient sensitive data. And this is where the weight of evidence tool is so useful, because it allows us a standardized way to combine those different elements to get an assessment of the certainty taken holistically. So area supporting evidence that we can add in to that assessment. So firstly, we have local investigation data. So this is some data that we collected around an investigation looking at the localized impact of Ilkley sewage works on the River Wharfe, and we did some additional diatom, macrophyte and phys-chem or phosphate sampling. That wasn't used for WFD classification, so we can, or the water body classification, so we can utilize that within the weight of evidence tool in that additional measured evidence section. We've also got sondes in the River Wharfe, looking primarily for as part of a bathing water investigations, but they can also be utilized for other purposes as well. And this is from this picture up in the top left, just shows the setup with the sonde. There you can see, I love these instruments. I have to say they're great. They measure real time, so every 15 or 30 minutes data from a number of different determinants. But the key ones that we're interested in are DO, and I've also got conductivity on that trace as well. So this is up in Grassington on the River Wharfe. And the light blue line is the dissolved oxygen percentage over several days in June a couple of years ago. And then the dark blue line is the conductivity trace. So what I want you to sort of draw your attention to is that diurnal variation in the dissolved oxygen. This is really indicative of basically photosynthesis happening excessively during the day, and we're getting supersaturation of oxygen up to, I think it's about 100 and 140, 150% in some cases. But then overnight, as those same algae and plants are respiring, it's dropping down to 60 or 70% overnight. And what's really nice about the resolution of this data set is you can see with the conductivity trace that's showing inputs of fresh water, which we use as a proxy for rainfall. So those dips that you can see, I don't know if you can see my cursor on this or not, but the dips are basically what we would consider rainfall events. And you can see when you get those rainfall events, the magnitude of that diurnal variation is dropping right down, and then it's increasing again as we go along. And what we think is happening here is a rainfall event's coming along and the filamentous algae is getting washed out. And then after the rain has passed and the sun comes out and we get hot, dry weather again, those filamentous algal mats start developing again, and you start getting that diurnal variation again quite quickly. So it's really nice to see that resolution of data and start to read what's actually happening in the river. I've also popped a QR code on there if you can try using it if you want on your phones. That should give you a live link to the current sondes and telemetry out on the River Wharfe. So it's just, again, a nice example of how we can share data in these sort of collaborative partnership projects. I can't see anyone's got their phones out, so I'll move on it. Can always come back to it. We also have some photos of algae and algal blooms in the river. This could easily be from citizen science. This is actually from one of my colleagues who was out doing some other work and taken off a bridge on the River Wharfe. But it just shows the power of a time and date and location stamped photo, and we can feed that into the supplementary evidence section of the weight of evidence approach. And then obviously there is the citizen science, and I know Rick has covered this extensively in previous webinars, so I won't go into too much detail, but we have used the data collected as part of the iWharfe project to feed into that supplementary data part of the weight of evidence assessment, and particularly the work that Professor Rick Batterby and co have done on looking at nutrient levels and also diatom results down the river. So this was a piece of work basically taking a snapshot in a single day from source. So all these are different sites running from right the headwaters on the left all the way down pretty much the confluence on the right. And what you can see is that step change around with the input of Ilkley sewage works and then subsequent sewage works coming down the catchment. So the top one here is phosphate, and you can see higher phosphate levels further down the catchment. And this is supported with the diatom data. And what's really nice here is obviously we've got the WFD classifications denoted by the coloration of the bars. You can see that shift from high to more good moderate status in that lower section of the river from Ilkley downstream. But we've also what has been looked at here is the community change, which is really interesting and shows that shift to more nutrient tolerant taxa, your naviculars and nichias coming in, which is really useful evidence, which we're able to put in as evidence from third parties or historical data that indicates eutrophication problem within that evidence strand. So this is the outcome then. Plugging all those different data sets through that weight of evidence approach, that decision support tool, these are the outcomes that come back. So despite the macrophytes failing at the top, the other evidence would indicate that there isn't yet a new eutrophication issue right at the top of the catchment. But as you move down, you can see sequentially all those evidence start to combine and pull together. And by the time we get to the lower two water bodies, we're very certain there's a eutrophication issue on the wharf. And this evidence and the fact that the lower water bodies were very certain was the evidence base for the EA submission of the lower wharf as a candidate sensitive area under Urban Wastewater Treatment Directive regulations to DEFRA. And the outcome of that was that it was officially designated earlier this year, and the link on the slide there will take you through to all the different maps of England that you can see all the new designations. But the one for Yorkshire is here, and you can see there's the wharf. So it's a new sensitive area in Yorkshire, and what that means is Yorkshire Water will now be required to install phosphate stripping at qualifying, and that's the larger sewage works that discharge into that sensitive area during the next round of investment under the asset management plan cycles. So what I would say is hopefully that gives you a real world example of how the Environment Agency uses citizen science data to supplement our own data sets, and shows when combined it can be a really powerful evidence base that drives significant investment in infrastructure by water companies, which ultimately improves our rivers, which is the outcome we all want. So thank you from me, and I'll hand over to Martin.
Thanks, Dave. Let's see if we can get it working. Hopefully it's a seamless change in slides. My talk will be looking at the use of data to assess eutrophication, but especially how we've been able to bring in citizen science data to supplement our evidence within the Wye catchment. Although we do have a weight of evidence spreadsheet, we haven't actually used it in this example of the work that we're doing. And it's mainly due to that we're looking more at agricultural, rural issues rather than the water companies, although we have worked with water companies previously and using the weight of evidence for PR24. So let's set the scene for you. As you can see on the map, hopefully that the Wye is actually a cross-border water body. So it starts in the Welsh Hills near Plynlimmon, a similar source to the River Severn, flows through Wales till we get to the border with England at Hay-on-Wye, and then it comes through Herefordshire, through to Hereford and down towards the estuary. So we, as an Environment Agency, are obviously mainly interested within the English section, but we have worked very closely as well with our colleagues in NRW as well. The River Wye is a SAC, so a special area of conservation. And as you can see, these are the pink areas on the map. So it's the main River Wye, but also parts of other tributaries as well. The SAC main features for its designation are its iconic fish species. So it's the Atlantic salmon, as you can see here. You've also got your twait shad. All lamprey species are present within there, and it's also designated for bullheads. It's also designated for its white-clawed crayfish population. It's also got a thriving otter population, and also for its water crowfoot population as well, its ranunculus. It is very high profile at the moment, and this has been heightened, especially with TV and social media coverage at the moment. So we do have issues within the catchment. The SAC is at the moment in unfavourable declining, as assessed by Natural England in Herefordshire. And these is due to excess nutrients, excess sediment, algal blooms, impacts of climate change, and invasive non-native species. It is mainly a rural catchment, and from our modelling, we've been able to show that actually 74% of the phosphate pollution within the rivers is deemed to be coming from rural land use. While around 20, 21, 22% is from sewage treatment works. We have a large problem with soil erosion within the catchment, very sandy soils. We get a lot of leaching of phosphates from the soils, but also just actual sediment going into the rivers, as you can see in the photo at the bottom here of a rather cloudy looking river. We've also seen a large increase in the amount of poultry units within the catchment, and Lancaster University have, through their Refocus team, have undertaken research on phosphorus movements within the Wye catchment, and they've seen through this that in the last 5 years there's been a large increase in the amount of manure produced within the catchment, and this has mainly been down to increases in poultry units. We've also seen an increase in maize production as well, which just also adds to the sediment problems. And then we've also been seeing algal bloom problems, similar to, as Dave's mentioned, within the Wharfe catchment. So we need an evidence base to confirm what these problems are and to find the most cost benefit locations for us to concentrate our resources as well to assess the SAC. However, within the EA, we only have a finite resource, and the River Wye is just one of our 10 WFD management catchments that we have to monitor. So although we're able to produce or look at quite a large area, and we do put the Wye as one of our focus areas, we do obviously only have a finite resource. So while we've been able to get a reasonable monthly water chemistry program in, and that's what this map is showing, the locations of those around the Wye catchment, we are also able to do some macroinvertebrates, mainly for WFD classification, but also for some water resources investigations. We have a principal salmon fish program, and we also look at diatoms and macrophytes, mainly for water framework directive and assessing nutrient pressures. We've been able to get some sondes in, and we also are able to start a few investigations using RAPPER, which is the Rapid Assessment of Periphyton Ecology in Rivers, or as sort of in layman's term, it's assessing algae on the riverbed. And then we also do some algal water column sampling as well. As I said, the Wye is a focus area for us, and we are trialling varying ways of using citizen science data to complement our work, due to the high amount of, well, the large area that we've got. to cover. So we've helped fund Cardiff University to set up the Wye Catchment Partnership. And by coming together under the Wye Alliance, we've been able to create a large group of citizen science people to be able, volunteers to be able to go out and collect data. So Cardiff University helped set this up, mainly with the help of the Environment Agency and the Wye and Usk Foundation, although also more recently NRW have been involved as well. It was great to get Cardiff University to help with the program design and actually doing some training materials as well for the citizen scientists. And it really did make a huge difference starting up the program. So these are the main citizen science groups that we have within the Wye Alliance. So we've got Campaign to Protect Rural England, Friends of the Lug, Friends of the Upper Wye, Friends of the Lower Wye, and the Wye and Salmon Association. As you can see, there's a really good spread of sites where they're actually going out and collecting samples. At present, the groups are mainly concentrating on basic water quality parameters such as phosphates, nitrates, assessing nutrient levels, although they are starting to show some interest in collecting other sorts of data as well. All the data is going on to EpiCollect, and that did start off as going under separate groups, but now we've been able to, under this Citizen Science Alliance, been able to get it under one group, which makes it a lot easier to be able to get the data out, and it's all fairly consistent in the way that it's recorded. So what are we doing with the data? The EA produce regular reports looking at the data from our monitoring, also using the citizen science monitoring, and also other third-party data to help build an evidence base, which helps us to focus our resources on reducing phosphorus entering the rivers. So these are two graphs, two maps, just to show you roughly where some of our monitoring was from our 2022 programme. As you can see here, we do have our algae sampling, and then we also have our sond locations. They're fairly well tied in. We've got the main River Wye flowing down here through the system, and that's where all our algal monitoring was concentrated on within 2022. And then we used our sonds program, not just within the main River Wye, but also on some of the tributaries as well. And this was really helpful for us to get a better understanding of what was happening further around the catchment. And all our sonds, although had the basics of the dissolved oxygen, temperature, conductivity, turbidity, we were also able to add chlorophyll on, which was really helpful for us to assess algal blooms within the water column as well. So there's just a few bits and pieces of information that we were able to glean from some of the surveys. So from our Wye wrapper surveys, so we're looking at that benthic algae population, we were able to see that actually as we move down, we're getting more at risk. So these are categories of where they are at risk of eutrophication. And generally this was at Hay and Wye, and then as we move further down the catchment around Hereford, we can see we're getting a lot more inputs, and especially during the summer months, we're getting an increase in those algal populations that are more tolerant to eutrophication. Also, with the help of the sonds, we found that the main algal blooms were tied into increases in water temperature, and that the main River Wye itself, the water temperature is much warmer than it is in its tributaries. And this is just sort of quite important going forward, especially trying to understand how we can help improve things within the Wye, because we're also seeing that these high temperatures are impacting the salmon population as well within the Wye. And especially during 2022, we had a reasonably large number of salmon reported, which were dying during the periods of the high water temperatures. So here we've got an example of where we're using not just our SOND data, which is the graph at the bottom. We're also trying to tie this in with our citizen science data. So as you can see here, we have our two SONDs here down above Hereford and below, but that's quite a large area. What we did find at this SOND down here, which we didn't up at the top here, was these increases in ammonium. But what we were able to do was then look at the citizen science data that our volunteers had collected around here, and the upstream site is this one along the bottom, and this was showing very limited impacts of phosphates through that time. However, the downstream site of Hereford here was picking up these high phosphate levels, and they tied in reasonably well with when we were seeing these ammonium peaks as well. And so that has furthered our work and our investigation onto this year, looking at some of the CSOs within Hereford itself, as we think that may be a potential source. We've also used GIS to visualize our citizen science data. We've got a couple of examples here of where we've used a heat map to try to show where the greatest issues are. For us, it wasn't surprising that the lug was looking as one of the hot spots, but we also found, especially for phosphates, is that in this one here, this is the River Frome coming through here, or Frome, and actually at the top of this, it was already starting with high phosphate levels. So we're already seeing that we need to sort of start focusing within this area to try to tackle what the issues are here. We're also seeing other areas where we're getting quite large amounts of phosphates coming in from certain tributaries as well. And all this information is leading into us doing further work on the ground with our agricultural officers and trying to make improvements. Nitrates wasn't so many sort of real hotspots shown. It was more of a consistent throughout the catchment. And just to say here, we also tried to make sure that we were using all citizen science data as well. So some of our groups were using the hanometers, while others were using the Lamotte strips. And while we didn't want to not include the Lamotte strips, we obviously feel that we're getting slightly better data from the hanometers, but it was really good to actually see how they fared against each other in certain locations where they're close together. And this is just to show that we weren't just using our data and citizen science data, we were also bringing in third-party data. So the map on the left is EDM data from Welsh Water in a heat map format. This is to show where the main discharges, amount of discharges that were going off in the catchment. Unsurprisingly, there was a higher frequency of discharges within the Herefordshire area, but it's also picked up really other areas which we might not have noticed previously, which need further investigation going forward. And then we've also brought in Chrome land use data maps from CEH as well, and we've been able to analyze these through time to see how land use change has altered things within the catchment. And within these maps, we've been able to see over the last 10 years that there's been a real increase in the amount of maize production within the catchment. With maize and potatoes on these sandy soils, this is a real problem for getting a significant increase in sediments running into our watercourses. However, just to say, it's not all a bad doom and gloom picture. Actually, the macro invertebrate community in terms of biodiversity has got very high conservation value. So these are just a few of the species that we've got here that are deemed as very high conservation value that we regularly find within the main River Wye. So we've got our riffle beetles, the Rhyolus nitens and the Stenelemus canaliculata. We have the mayfly, Potamanthus luteus, where this is a real stronghold for the Wye, although we have now started to see this moving within to the Severn itself as well. We have the other mayfly, Ephemera lineata, and also a CaSTCo Leptocerus interruptus. And all these are really good indicators of high water quality, which you can also see here in the graphs looking at our WHPT scores, which we use to assess the impacts of organic pollution. And for ASPT, so that average score per taxon, and also the number of taxon. The red dashed line is the good moderate boundary, and as you can see, generally the majority of the sites that we were looking at within this investigation were all above the good boundary. A lot of them within high. What we did notice a little bit more this year than previous years was this was from 2022, and we were definitely seeing a reduction in the autumn samples at all our sites. And we think this has been the impact, measuring the impact of that really hot, dry spell that we had during 2022. What we do try and do is try to make our data easy to read and give some of the main points out so people can very quickly see what the issues are rather than having to go through a full report. So we've tried producing these visual summaries just to try to get the message over nice, simply and quickly, showing what we believe the main sources of algal increases are within the Wye, mainly due to agriculture, but also those extreme temperatures and rainfall events. And especially we've seen the rainfall events during low flows during that dry summer when we were getting those summer storms. They were causing real issues within the rivers over short periods because we were getting a lot of rainfall flushing nutrients into the rivers, and also the impact of CSOs was a lot worse during those periods as well. We also highlighting the issues with the fish and the algal, but the fact that our invertebrates are really good, and then recommendations going forward. But what is really pleasing at the moment is that our citizen scientists are looking at their own data as well, and they've been able to create this really great YViz platform so they can view their data quickly and easily. And it also makes it a lot easier for us to get that data and have a look at it as well. And they're also starting to produce reports on small catchments as well, actually looking at that data and actually, highlighting issues that we maybe wouldn't have been able to get to look at with just our data that we've got. So this is being a really useful collaboration with the Wye citizen scientists. Just as a final plug as well, is that we do have a Wye website, and I'll just drop a link to that in the chat. And this is a really helpful way for us to engage with people. We've created this so we can share the information that we're collecting, so our data reports are in there, but it's also a really helpful place for citizen scientists to actually come together, find useful resources within, and find a bit more information of what's going on within the Wye, because there is so much going on at the moment. So that's my sort of wrap up of the Wye. So as far as the wider sort of presentations that we've been given today, we thought it would be worth just having a few conclusions to wrap up and finish. And I think one of the key things is, especially from Dave's talk, is consideration of how best moving forward that we can start utilising citizen science in that weight of evidence process. We think that all data is really useful, but we maybe need to add some sort of weighting depending on the scale of the evidence that's being collected. We also hopefully are showing that it's important not to just focus on one strand of evidence. We've got to look at the larger picture. We've got to start looking at the whole system to see what is actually happening. It gives us a better understanding. We also believe there's real promise of development of using Wrapper or something very similar in the approach of citizen science. And I know Bill Briley has been looking into this, and we've got research students looking into a way that this can be done for citizen scientists. And this is be a really good, rapid way of citizen scientists helping us to add data and eutrophication impacts. And then the metadata hub is really important. And this could help identify different data on nutrient impacts, hopefully going forward. And finally, just that, you know, citizen science data can be an integral part of that evidence base going forward. We really do value that. And the EA is invested in using citizen science data. Through CaSTCo, we've had funding to make sure that nationally we have a national citizen science advisor now, and then in each area as well we have a citizen science advisor who is real, and they are making a real difference on helping us use the data, but also getting that interaction with citizen scientists and being able to get two-way discussions. And within West Midlands, we've seen a real progress, and we've seen establishment of a number of groups within West Midlands of citizen scientists. So, and that is the end of the presentations. Any questions?
Thank you both. That was really informative, and I feel like a great illustration of definitely the ecosystem-wide impacts of getting that wider data. Really cool to see. Thank you. I think we've had a few questions in the chat, so I'll run through those, but anyone else who has questions, please put your hand up in the meantime, and then we'll come through to you. So Amanda has asked, can we have that QR code again? Or Dave, if you're able to put the link in the chat or something, that would be awesome. Thank you. Cool. And Amanda also has another question. Amanda, you're welcome to ask these yourself if you want, but I'm happy to read them out. But is there a way to remove phosphate at source rather than end of pipe removal? So I don't know who would like to come in on that.
I'll let you do that, Dave.
Thanks, Martin. Well, obviously our specialism is on the ecological response in the river, so necessarily the solutions. But what I would say is there are a number of approaches to reducing phosphate inputs to rivers. Obviously our talks are just covering, well, hopefully we cover both actually, because there's obviously the sort of sewage infrastructure inputs, which was covered in my case study on the Wharfe, but that's not, you know, forgetting there's also contributions from multiple other sources, from agriculture, septic tanks, you know, a whole range of other sources too. So any solution to get sort of system-wide change, as Martin's alluded to, would need to address all of those inputs and its different approaches to different inputs, basically. So this was just focusing very much on one specific issue, which is continuous discharges on sewage for the Wharfe. But obviously Martin's covered that more agricultural diffuse pollution approach on the Wye, and what we've not covered are those other sources as well. So yeah, there's a lot of different approaches needed to crack this particular nut of nutrient pollution.
Yeah, I don't think I can add much more than that, really, Dave. As you say, it is depending on the catchment. there are various ways that we can look at it, but we do need to. But we are sort of ecologists, so we leave that sort of discussion to our other parts of the agency or other regulatory organisations.
Super, thanks, Boo. I think quite a few of these questions in the chat have been answered by other types. Do you see any value in a centralised approach to storing and sharing citizen science data, like a national database rather than local catchment data sets?
From an environment agency perspective, well, something we've been sort of pushing through the Wharfe project, and then going forward has been the development. I think the name's changed now, but basically looking at the metadata of what data is available and where. And I know the Rivers Trust has been building that system, and that will be so helpful and so useful, a one-stop shop to see who holds what data on, where in the catchment. I think actually having a central sort of database for all data is going to be quite a difficult thing to pull off, but that's why we've gone down the metadata route, because that can bring in, we're not just talking citizen science, but other water company data, consultant data, academic data, statutory data from the different regulatory bodies, and show who holds what in what area. And that's the sort of format we need, so that we can go into a water body, particularly for something like the weight of evidence assessment, and say, okay, what relevant data do we have in this water body? And then if we need, sometimes there might be a link through to it, on that metadata hub, but other times you might have to just have a contact details of how to get hold of it, and that allows for different approaches and different scales of organisation, I think. So.
Thanks. I'll go to Tristan next, as you've had your hand up for a while, Tristan, so please go ahead. Oh, I can't hear you, Tristan. Sorry. No. We'll go to another. Oh, there you go.
Now? Yeah, you can hear me now?
Yeah,
okay.
Yeah.
Sorry about this. I've been having problems with my headphones working properly. Anyway, right. Tristan Hatton-Ellis, Natural Resources Wales. So I've had a long-standing link with Water Framework Directive tool development and classification over the years. And I've also had a bit of involvement. I haven't used the Rivers weight of evidence tool, but I've used the Lakes one quite a lot. And one thing that has struck me while using it, and probably applies to the Rivers one as well, is it relies on there being quite a lot of data available. So to be certain about stuff causing a deterioration, you need quite a lot of classification data. And obviously we're not, that was fine maybe 10, 15 years ago when we did a lot more monitoring. But that's not really the case anymore. And so I think a potential weakness with the tool, with using the tool these days widely, is going to be that we don't have that. Now, I know that there is scope for bringing in the citizen science there, and that bumps the certainty up a bit, but my memory of the architecture of the tool is that you could never really get it high confidence using the citizen science data, because the tool doesn't consider the citizen science data to be as robust, rightly or wrongly. So I guess that's something we might want to have a think about in terms of how we look at the tool. I also think the other thing is how to interpret the outputs of the tool, because, and this is a little bit of a thing I have with the approach we take to river ecosystems. We kind of view river ecosystems as being something that just responds to water chemistry. So you put some phosphorus in and it changes, it goes up, something goes up or goes down, and it's not like that. So actually you sort of, or it's not like that straight away anyway. So, we saw that there was that horrible photo of all that algae on the wharf. Now, it might well have been that the phosphorus status was still at high status or good status, because what happens first is when you put some nutrient in, the phosphorus, the algae go great, and they grow a bit and they soak up all that phosphorus. And particularly if the flow is low, your phosphorus concentration might stay low, but you've got a load of crud in the river. Now, So actually, there you've got the biology is affecting the phosphorus. It's suppressing the phosphorus concentrations and keeping it down. And it's only once you put so much phosphorus in that the biology, however much algae you have in there, you can't possibly absorb it all, that you actually start to see those increases. So I think there's some, that's an example. There's loads of examples of that sort of thing happening all the time. So I guess what I'm saying is, when you put up that table of the different tools and how they align, they're never going to. They're always going to show slightly different things, and it's going to depend on conditions in the river and all that kind of thing. So I think. I think it's good to see this as a multifaceted problem rather than something that's just a linear thing where we say, okay, we measure a load of phosphorus and that phosphorus immediately results in something happening. It may be that something has already happened, or it may be that there's other things. And I think that's where citizen science can really help, is maybe you guys can do more wrapper surveys, or maybe you could do some more river habitat surveys, some of these wider things that will give us a richer and deeper understanding of what's happening in the rivers at different spatial locations, and to fill in some of those gaps as well in where I don't think we can necessarily, so a lot of the classification tools, I think asking for diatom surveys is going to be a bit much, but there might be ways to do a cut down plant survey. I don't know. There might be certain things that could be looked at.
Yeah, I think we both agree, don't we, Dave, with that? And I think going forward, we would like the weight of evidence to be looked at. Going forward, that might happen, but I don't think at this present moment, is it, Dave, that there's.
I think there's, I don't know what the long-term plans are going forward, but you certainly make a good point, Tristan, about the sort of monitoring landscape changing and making sure that tools reflect that in terms of the data used, and the other points. I absolutely agree. The nuances and the variables that go into that response. Hopefully some of the data we showed, just the time since the last rainfall event, the level of shading, the level of impoundment in the watercourse. There's so many variables that come into that assessment of how that river system is responding to a nutrient load, and how much capacity it has to almost buffer it in different scenarios as well. There's loads of considerations, and that's why that decision support tool is so helpful, because you often get contradictory data, and I think that's something we just have to live with in that field of looking at nutrient response.
I think for PR24 we did put in a number of local investigations just to try to build up that evidence base as well around certain sewage treatment works which we felt we needed that evidence for. So.
Yeah, definitely. I think the other point is that just because you have somewhere that's showing as no risk, or even low risk rather, or even no risk, doesn't mean you shouldn't do cheap and cheerful stuff like fencing or working with farmers and stuff, because it'll still be contributing a bit, and it may be just that you actually catch a problem before it starts to show in the river. Whereas obviously if you're going to throw something expensive and controversial at it, like 10 million on new sewage treatment works or something like that, you would require high certainty, wouldn't you?
Fab. I'm afraid we've run out of time. That was a really, really great session. I've got a few more questions in the chat, so I'll send them over to Martin and Dave afterwards if you're happy to answer them offline, and then we can share those with everyone as well. But thank you so much, Martin and Dave, for presenting. It was super informative. Really cool to see those case studies as well. And hope to see you all at the next webinar, whenever that may be, TBC. So thank you all.
Thanks, everyone.
Thanks very much.
Thank you all.
Cheerio. Bye.
Bye.
Cheers. Thank you. Bye.